Hello, I'm Russell Hope. Welcome to the AMR lectures. I'm going to talk to you today about AMR data science challenges from a One Health perspective. The learning objectives for this work is to have a little bit of an understanding and appreciation for antimicrobial resistance. We'll examine some of the examples of digital gaps and challenges in AMR, and we'll look at the key questions at the end. So here's some suggested pre-reading to get you a little bit familiar. With antimicrobial resistance, where you use an antimicrobial, you can expect at some stage there to be resistance. So here you can see a timeline of drug discovery on the right-hand side, and on the left, you've got the occurrence of resistance. So you can see penicillin resistance coming in shortly after application of penicillin. So you can see that fairly quickly after introduction of an antibiotic, we can expect to see resistance emerging. And this has followed over time. And as we develop new antimicrobials to overcome the resistance, then you can expect to see resistance to that new antimicrobial. And here you can see some developments within Staph aureus, with penicillin resistance, methicillin resistance, vancomycin resistance and intermediate susceptibility, and linezolid resistance in 2003. And you can see similar things for enterococci and also for tuberculosis. Tuberculosis is particularly problematic because the therapy durations are so long. So it's easier than some other agents to see resistance development. And you can often see stories in the press around antimicrobial resistance. Here's one from a few years ago around resistant gonorrhea. And again, in some of the gram negatives, isolates becoming extremely resistant, very few treatment options. So antimicrobial resistance is a One Health issue, and you can often consider One Health involves animal health, agriculture, the environment, etc. And these can seem a little bit distant from our everyday lives. So here's an example from the One Health report where we're looking at a One Health AMR issue, but a little bit more closer to home. So here you've got a pet owner feeding their pet raw meat. Raw meat's been shown to be highly likely, about 39% of cases had an E. coli that were resistant to at least one antibiotic. And then because you're not cooking the meat, which normally removes much of the threat of transmission, then transmission is much more easily presented, and there's a number of different routes where you are seeing contamination flow from the dog to the environment in the house, to the environment outside, and ultimately transmitting to the owner. AMR is not just a concern for where we are now. In the UK, we've seen some fantastic reductions in some of our resistant infections. For instance, MRSA bacteremia, where we've seen large reductions in the 2000s. But we want to try and maintain those gains. We want to try and stay ahead of the curve. There are plenty of instances where this isn't the case. And so there's been a big push from government and supporter of the AMR agenda and try to highlight the risks. And this is because if we see AMR develop, it really puts in jeopardy some of our techniques in modern medicine, where things like routine surgery would become quite problematic in some instances. And so to avoid this threat, action needs to be taken. And the UK and a lot of countries throughout the world have generated strategies to try and mitigate antimicrobial resistance. Because AMR isn't just a problem in this country, it's a global one. And here you can see some estimates of the global burden of AMR. So both a large number of deaths and in comparison to things like AIDS, breast cancer, malaria, etc. And then some projections here up to 2050 with 10 million deaths. And alongside that, there is a substantial economic cost as well. So along with those national action plans, the UK government also released a 20-year vision for controlling AMR. So those 5-year national action plans, they fit within that 20-year vision, and there's various different ambitions within that. So looking at key themes is to reduce the need and unintentional exposure to antimicrobials, looking at optimizing the use of antimicrobials, because it's not just about what antimicrobial you use and when you use it, but how long you use it for example, and investing in innovation. So, remember we saw that slide at the beginning where we saw new antimicrobials coming in and then resistance being generated. If we don't continue to innovate new ways of controlling infections or new antimicrobials, then we can start to fall behind the curve of resistance that's generated, and we'll have fewer and fewer effective compounds. So let's look at some AMR examples. So this is Candida auris. So this is a fungus. And we saw a number of UK outbreaks between 2017 and 2019, both local transmission and new importations. Due to travel between countries, etc., this infection spread globally, and you can see from the graph at the top here where it's been detected, where we're seeing outbreaks, and in some cases it's become endemic. Now, during COVID-19, the numbers fell a bit, but more recently we've started to see numbers increase. Why is it an AMR issue? Well, in this case, C. auris is resistant to the azole antifungals. So you can see the minimum inhibitory concentrations highlighted for a selection of isolates, and they're all in that resistant red area. So that's for fluconazole. Used fungi here as an example because a lot of data you will find on AMR is on the bacteria, not the fungi. So it's nice to have a fungal example. It's even rarer, I think, for antivirals. So that's one of the data concerns. So here's some example of some resistant bacteria. So this is 2 different strains of Klebsiella harboring carbapenem resistance. And carbapenems are some of our last resort antibiotics used in some of our illest patients. So carbapenem resistance is very serious. And so here you can see in orange, we've got spread of carbapenemase, a carbapenemase called KPC, which is Klebsiella pneumoniae carbapenemase. So you can see how it's spread globally, and likewise on the blue trend or arrows, you've got New Delhi metallo-beta-lactamase, both spreading in Klebsiella, but different carbapenemases, and both with international spread. So the use of the antibiotics generates that spread of bacterial resistance, and it does that because it kills the susceptible population, and what you're left with is the resistant population. So this diagram summarizes that. That's fairly straightforward. So there's a number of biological processes that facilitate this. So we have clonal spread, which you saw on the diagram of those 2 carbapenemases spreading, but you also get spread of the mobile genetic elements that can harbor the resistance genes, such as plasmids, for example, although this isn't the only mobile genetic element. And you could have mutation where antibiotic resistance will develop de novo. The mobile genetic elements give us particular concern. So here's a picture of some bacteria sharing some DNA. So a plasmid is being exchanged here. And so not only can you get this clonal spread of organisms, but they can share the plasmids and other mechanisms. But the plasmids in particular can be problematic because they tend to be associated with multiple resistance mechanisms. So you can have a plasmid with many different resistance genes on it, conferring a antibiogram that will give you resistance to a myriad of different antibiotics. And that disrupts that kind of selection pressure. So you think when you use one antibiotic, you're going to get resistance to that particular antibiotic. But if the resistance mechanism is carried on a multi-resistant plasmid or a mobile genetic element with a number of different resistance genes associated with it, you will actually not only select for resistance to the antibiotic you're currently using, you'll also select for resistance for the other antibiotics that the resistance genes confer resistance to. Another element to this multi-resistant plasmid spread is that it doesn't need to be the same species that the plasmid moves into. So you can see a multi-resistant plasmid from a Klebsiella move into a Serratia or an E. coli, for example. And so this will present as a number of patients having infections caused by different species, and therefore you wouldn't naturally think of it traditionally as an outbreak. Certainly, they're not the same species, they're definitely not the same clone, but the resistant element that has been shared is the same. So in actual fact, you do have an outbreak on your hands. And this means that to get this level of granulation and to be able to capture these kinds of outbreaks, you need very detailed molecular work that is not only looking at the clonal lineage that you're dealing with, but also characterizing those mobile genetic elements. And that invariably relies on you using whole genome sequence techniques. And these are not commonplace. So this is a real data gap at the moment, although it is improving. So that moves us on to discuss some of the data gaps. So during COVID, the public, got much more familiar with looking at data, which is fantastic. And their appetite for looking at health-related data and using dashboards has increased. And that's a good thing because we want to try and promote knowledge about antimicrobial resistance. So here you can see some examples of different interactive dashboards. We have some of these for antimicrobial resistance, but there's definitely a need to continue to develop these and make sure they are tailored for the right audience. And part of that is about engaging the public, but it's not just about enhancing the knowledge that the public have about antimicrobial resistance. We can actually get the public to actively participate. So there have been a number of examples of public citizen science programs where they've been able to help identify resistant organisms or help us identify possibly cancerous cases, etc. And so there's definitely more that can be done in this area with AMR, even if it's just getting members of the public to take swabs so we can gauge the true or gauge the better picture of the level of prevalence of antimicrobial resistance that we're seeing in the general population. So here you can see some data of some of our mandatory pathogens that we track in the UK. So you'll recognise some of these like MRSA and Clostridium difficile. For example, these are some of our more well-established healthcare associated infections. And you can see here the trends before, and then you can see a dip, which was during COVID, and then a recovery. Now, this data is currently collected quite manually. If you want to construct this information via electronic means, then you have to merge lots of different data sets together. And that data is available. So we're definitely entering the realm of big data with these infections. And there is a lot of data out there. However, it is not always easy to find out what's available. And once you have found out what's available, accessing the data can be difficult. So often the data isn't open and you have to apply for access. And that can be a very long-winded process to do. It's necessary because some data is identifiable, for example, so you could identify an individual from it, and there is understandably a lot of protection around that data. We don't want to expose people's personal information, but there is ways of pseudonymizing data. So one of the challenges around big data is to make it more discoverable and have a safe space where researchers can interact with it and use it in their work safely. And to this end, one of the grand challenges we're looking at with the Digital Hub is to look at the foundation needed for a One Health trusted research environment, which will link human, animal, and environmental data together and help that kind of data discovery. This can build upon some of the prior work that's been done on this relatively recently. So the Pathogen Surveillance in Agriculture, Food and Environment Programme, which is run by the Food Standards Agency, is a cross-government organisational project. And it was all about developing methods for looking at AMR in the agri-food chain and demonstrating sort of proof of principle. And one of the parts of that work was to look at a One Health surveillance system, so sort of like reporting dashboards, etc., as a discovery piece. So this and the EPSRC grant can build upon this. So as part of the discovery, we had to look at what were the sources of AMR data we wanted to collect and who the key stakeholders were and what they wanted from AMR data. As part of this process, we established things called user personas, which is essentially a picture of the kind of users you might have and their different requirements for using the system, because there's a number of different use cases that will develop from this. We also looked at existing data flows. So part of that work about finding out what data is available, not only what's available, but where it's stored, how it's stored, etc. So that we contacted a number of different organisations throughout the UK and looked at how and where they stored their AMR data. We held workshops, etc., and gained user needs and requirements. So when you're looking at generating an output, you need to know the kind of outputs that people want. And so this was done as well. So this was the discovery work around the main functionality that would be needed from this environmental system. As part of this work, we also built a AMR reporting system. In this case, it was just showing some environmental data from some river water samples, but it gives you the kind of idea of the visualizations you could create that are relevant for AMR data. So here at the top, you can see a bit of a bar chart, which is showing you different minimum inhibitory concentrations and the number of bacteria or samples that fall into those. So it gives you a good idea about the proportion of susceptibility. You can see green is susceptible, red is resistant in this case. And then on the bottom, you've got a violin plot showing you the concentration of different compounds within some of the water samples. So it's not, when you're looking at AMR surveillance, you won't necessarily just be monitoring antimicrobial use and looking at resistance. You may also be looking at levels of antimicrobials in the environment, but maybe also things like heavy metals, etc., which can be co-selected on those multi-resistant plasmids we were talking about earlier. So sometimes you need to look at more than just the antimicrobials. One of the big challenges in recent years has been healthcare utilization. So during COVID, we saw a big increase in remote consultations. So here you can see that the odds of 1.23 odds ratio of an adult being prescribed an antibiotic without being seen face to face. So that went up during COVID and it hasn't come down. I think we're not really going to see that change now. And in addition to that, in England, we have rolled out the Pharmacy First services. So this is where, for the conditions in the table, you can see there, you can go to a pharmacy and the pharmacist can assess you via a selection of criteria and decide to take action based on that, which can include giving you a prescription or referring you on to your GP. But again, it's another avenue for patients to receive antibiotics without seeing their GP. And some of this prescribing will obviously occur within private companies as well, which means that you have a source of data which is held by the private sector. So another example of where data can be, data on the same, so prescribing in this instance, can be stored in a number of different places, a number of different organizations, a number of different sectors, etc. So why are we concerned about keeping an eye on this? Well, we have seen prescribing was coming down as part of our ambition to reduce selection pressure, but and that kind of bottomed out during COVID, but we've subsequently seen it increase. So we need to keep track of these changes in healthcare utilization. And there will be a lot of scrutiny for the Pharmacy First activity in England. It has been used in Wales and Scotland as well, and has been very successful. So I don't perceive us having any real problems in England. So we've got this smorgasbord of data that's available. You can house it together in trusted research environments, etc., but trying to make sense of it is really quite difficult. And results I've been showing you so far are all sort of like national level results, and it's good to know what that trend is. But what does that trend mean for an individual patient? So there is a need to translate all that knowledge and pool all the data you have available into tools that can then guide a clinician as to the kind of therapy and the duration of therapy that they might want to use for their patient. And here's an example of a individualized patient data tool that Imperial College have been working on. And things like AI will hopefully allow us to make sense of this complicated data. So you've got your data coming out from a hospital, from point of care tests, from academia, from the environment, from One Health. You've maybe got some, not true environment, but wastewater sampling going on. And you may want to consider things outside of it like climate change, etc. One of the things you need to do, even if you have all the data in separate tables, etc., in your fantastic trusted research environment, you're going to have to link them together in some way. And this takes an awful lot of compute. And one of the challenges we find is when we have data scientists coming to work on these different data sets, they will often approach data linkage as something new. And although they can learn from what has gone before, there will be a lot of finding their own feet, writing their own code, etc., adapting what's been done. So we wanted to look at how data linkage was done. And here's an example of a data linkage engine that we built over the last few years. And the idea around this is to enable linkage of some of our key data sets. In this case, this is all around human health, but it's taking things like the secondary generation surveillance system, which holds microbiological data, and linking that to hospital records in the hospital episode statistics, and death information and prescribing information, etc. So this is work we would have done before, but this linkage engine makes it much more straightforward to do. So it creates automated pipelines so that the data linkage can be scheduled. The scientist doesn't need to actually know how to perform the data linkage. And importantly, the data scientist never needs to be exposed to any of the patient identifiable information to link the cases. The software can do it all for them. They never need to touch it. So that allowed us to remove some risk for the system. And when you're dealing with these really large data sets, then often they're so large that it surpasses the kind of infrastructure that a lot of our data scientists had access to. So we needed to provide a system that was performant in terms of time, but also could scale both vertically and horizontally. And so for that, this system exploits things like containerized environments and code as infrastructure to make that happen. And here's an example of some of the data we were able to put together with this data linkage engine. So this is some COVID data you can see, and the COVID cases have been categorized by how soon after a patient arrived in hospital the COVID case was detected. The idea being the longer you've been in hospital before you've got a positive COVID sample, the more chance it is that you are exposed to COVID within the hospital environment, although it's not always so clear cut. So this data was all done using data linkage of big data, and this was done at the height of COVID. It was done on a daily basis. Something that wouldn't have been possible without the data linkage engine. So all the data we've looked at so far has been around data from infections. So you only get that once the patient has presented, is ill enough to present with an infection. And that means that you only find out about the infection once it's already progressed to that stage. What would be good is if we could have a steer of what was happening before you saw those more severe cases. And here's an example of where you can use the internet searches that individuals are doing to give you a heads up about an infection that might be spreading within the community. And this can let you escalate your surveillance sampling, etc. And this means that you can be more timely in your response. So there's definitely a need to look at that a bit more closely. And there you can see the microbiological data associated with scarlet fever, which is what the Google searches are tracking at the bottom. So that's some example of the sort of like digital gaps and challenges around AMR. So let's just review some of the aspects of that. So you've got data from many sources, and that can be challenging in terms of accessing them or knowing about them in the first place. So there's a data discovery element to that, and things like trusted research environments, etc., can help with that. Likewise, you've got issues with data formats. So with your trusted research environments, you can also standardize the way that data is presented, etc., because across the smorgasbord of data that's available, it's not going to be in the same format. In some cases, you will still be working with organisations where at least some of their records are still on paper. That's becoming increasingly rare, but can still be the case. You may be working often with skewed samples. So a lot of the data we're looking at is generated from routine microbiology, which is there to determine why somebody is ill, or reference services where people are sending in isolates because they think they've got an outbreak, or ad hoc research studies. And these are all done for different purposes, and therefore they may not be particularly representative for the question you're trying to answer. So you've got to consider that when you're using these various sources. And this is even worse when you're looking across One Health. So it's bad enough when you're looking at human health. Once you include a One Health agenda, it gets far, far worse. And you can expect things not to align, like geography or timings or the focus of the study, etc., except in some exceptions where you've got something that has been particularly tailored for your use case. We already talked a bit about the infrastructure challenges. So as you get these large amounts of data, that's a big challenge to just store it, let alone manipulate it or analyze it, etc. So we talked a little bit about the use of the containerized environments and infrastructure as code that we used for UID, but these are definitely some useful tools in dealing with these kind of infrastructure problems. It lets you move your code around and your storage around easier. So use of cloud computing, etc. there. Staff training is important. A lot of staff won't be familiar with dealing with these large volumes of data. They need to have access to languages like R and Python. They need to be able to query relational databases, so SQL is important. They need statistical analysis. Where you've got groups of people working on the same problem, or you've got code that you want to share, then using collaboration and version control software such as Git is critical as well. It is also essential for the infrastructure as code and being able to automate running code. So the UID example I gave you earlier uses Git a lot. And then for dashboarding, I showed you some examples of dashboards which were built with R Shiny, but there's also other options like Tableau, and the PathSafe example I gave you of dashboards that was built with Power BI. So there's a number of different dashboarding tools that are available, and getting familiar with these tools will allow data scientists not only to be able to present their data to groups of people, but also allow those individuals to go away and play with the data visualizations themselves, because they generally tend to be interactive. We need to make sure when we're building these different applications, etc., that we engage with stakeholders. We need to understand what they need from the data, where they think the gaps are, and the kind of way they want to see the data as well. So it's no good sharing reams of information with somebody, even if it's the information that they ultimately want, if they can't really get to grips with it easily. They need to be able to understand it relatively straightforwardly. Otherwise, it's going to be challenging for them to use it. And we need to make data as timely as we can, because data is for action, and you can't take action if you find out about something well after the event. So timeliness is critical. So let's look at some of the key questions. So the real thing we're trying to do is really stop AMR transmission. So we need to look at things like acquisition to infection risk. Does decolonization work? Certainly there was some fantastic work on decolonization around MRSA, but it's not applicable for all resistance problems. And certainly doing things like trying to decolonize the gut to reduce the risk of, say, an ESBL or a carbapenemase producer is much harder and hasn't seen so much success. We need evaluations to look at how we can interrupt transmission and then assess how effective that is. Likewise with new products, etc. We need to expand and educate the workforce in AMR, but also the tools that are available. When we're looking at interventions, it's not just about having that new intervention, it's also making the intervention that needs to be taken into account. It's not just about doing things like setting targets. And we can look at how AI and personalised approaches can help. We need to involve an economic impact of AMR because every action that we do has an economic impact. So it's good to demonstrate that this activity is worthwhile because that helps us secure funding for us to be able to keep this all going. And it's not enough to just have something set up. You need to keep on adapting it. You need to change. The antimicrobial resistance is not going to stay static, as it has demonstrated time and time again. So we need to be able to match pace with that. So some conclusions. So there's a need to try and bring that One Health data together as much as possible. Ideally, we'd start to get some global harmonization as well. That's a huge challenge. We need some new sort of like novel laboratory approaches, particularly around mobile genetic elements, so molecular diagnostics, etc., to be able to look and detect those plasmids so we can identify those multi-species, same plasmid outbreaks. We need things that could encompass the whole pathogen approach, so looking across food, sexually transmitted infections, healthcare-associated infections, etc., not just focus on bacteria, consider fungi, virus, parasites, etc. It does need to be across the board. There's a role for localized diagnostics and bespoke data development to retain information, but also we need to make sure we retain that data in public health systems. So yes, you need detailed information locally so you can take action, but you also want to know what's happening around you and how that may influence what's happening locally. So you may, for instance, want to isolate some patients if you think they're at greater risk of carrying particular resistance mechanisms. So to conclude, we need to harmonise global reporting across One Health. This is a huge challenge. Just doing it within one country across one sector is difficult. It's going to be even harder across One Health. We need to develop novel laboratory approaches, particularly around molecular diagnostics, so we can detect those multi-species, same plasmid outbreaks, for example. We need novel surveillance approaches, looking at things like wastewater to detect community prevalence, etc. And these need to be designed with this approach in mind. So we can do a lot with the data that's already available, but if the surveillance programs are designed from a One Health approach, they will be more robust. We need to encompass the whole pathogen approach as well. Looking at bacteria, but not just in humans, look at it in food, etc., and look at things like fungi, viruses, parasites, etc. We need to increase the role of local diagnostics and bespoke data development, but we want to make sure we retain also that more regional and national and global view of trends, etc., because they can also inform on threats that will affect the local setting too. There's a critical need for new antimicrobials and novel alternatives and the evaluation of those because, as we've seen, there's that constant pressure of antimicrobial resistance developing against the compounds we have available to us now. And there's a large role in this for data discovery, including citizen science. Thank you very much for listening. Here's a further reading list. So this just leaves me with my acknowledgements slide. So I'd like to thank Colin Brown and my colleague Lissa that contributed a lot to these slides. Also, a lot of the work that you saw in the slide set from the PathSafe Work Package 4 team and the UID team. But in general, this work and the kind of presentations you've seen throughout this course of lectures is not possible without the work of public health professionals, healthcare professionals, people working in animal health, environment, industry and academia, etc. It makes it all possible. And also thank you to EPSRC for funding this hub.